1, you can see the autonomic reflex arc. In this case, they have given how these two systems, the sympathetic and parasympathetic, you see the names on that page, those are the two divisions, how they regulate, in this case, blood pressure. So there are sensors on the blood vessels on your heart. You see the green buttons or the green dots. So these sense change in pressure, and then they send the signals to the brain, and from the stem of the brain you get signals back, and then your blood pressure, for example, is adjusted. If it's higher, it's lowered. If it's lower, it's higher. It's made higher. Okay. So this is a general schematic that they have given, but we will see how these things work. But before that, you need to understand what is sympathetic and what is parasympathetic division. So sympathetic division is the one division of the nervous system. Okay, you have the brain, you have the spinal cord, you have the spinal nerves, right? Or the cranial nerves. But some of these, after they branch, some of them, they make a grouping again. So some of them end up to be the sympathetic, and some of them end up to be the parasympathetic, and you will have both fibers reaching to your target organ. Okay, so the sympathetic division normally speeds up or increases the action, with some exceptions. Like if you see, it inhibits digestive system, but everything else it increases blood pressure, glucose level, alertness, heart rate, pulmonary airflow, blood flow to cardiac skeletal muscle. At the same time, it reduces blood flow to the skin and digestive tract. The parasympathetic division, it tries to have a calming effect, or it lowers everything down, with the exception of the same thing, the digestive system it stimulates. Okay, so sympathetic division is basically turning on, turning off certain things when it goes out of control. Okay, normally the nerves, the cranial nerves, spinal nerves, they operate whatever they have to operate. But sometimes things go out of control. Then you have further divisions that regulate and bring it to your normal values, whatever is going out of the normal range. So these two systems always work, they are always working at some level. They call it autonomic tone, like muscle tone. Muscle is always partially contracted, ready to go. So they call it autonomic tone. Then figure 15.2, you can see the difference between the somatic versus autonomic system. The somatic efferent innervation and autonomic efferent innervation. What is efferent?
Away from. Like away.
You remember afferent, association, efferent neurons?
Away.
Afferent taking the sensations to the brain, efferent, so it's motor. Okay, motor function is efferent. So these are all motor nerve fibers. So on the top you have the somatic efferent innervation. You see the nerve fiber originating in the spinal cord, and then it goes all the way to the target muscle, or whichever the voluntary site of action. And what is the neurotransmitter that is being released? We did the muscle action, right? Acetylcholine. So you see one long myelinated fiber, and then you see the acetylcholine in the synapse, and then it activates the muscle. But if you compare that with the autonomic, how many nerve fibers you see there? 2. Are they both myelinated?
No.
First one is myelinated, and the second one is unmyelinated. And on top of your dorsal root ganglion, that is close to your spinal cord, we have another ganglion they call autonomic ganglion. What is basically a ganglion? Collection of your cell bodies of neurons, the soma. That's why it looks big. Okay, so in that first synapse, it's ACH always. But the second synapse, where it gets to the target organ, It could be either ECH or NE. What does NE stand for? Anybody? Norepinephrine. Remember epinephrine, norepinephrine, these are produced by your adrenal gland located above the kidneys. And these two neurotransmitters, they also have other names. Adrenaline is called epinephrine, noradrenaline is called norepinephrine. Okay? So the fiber before the ganglion they call preganglionic fiber. The one that's after the ganglion they call it postganglionic fiber. I may have told you about presynaptic and postsynaptic neuron earlier, right? Same thing, but here you have the ganglion, so they call it pre and postganglionic fiber. And they have compared both the systems in table 15.1. And I want you to take 5 minutes and look at figure 15.4, which shows the arrangement of the sympathetic nervous system, and compare with figure 15.7, which is the parasympathetic nervous system, and use table 15.3. So you can kind of fold it halfway, that page 570, so you can see both side by side, the sympathetic and parasympathetic, and see what are the differences like. Do you see certain ganglia in one versus the other? How is the branching? Are there too many branches? Where are these branches coming from? Which part of the CNS? Okay, whether on the top or the bottom, which region? And if you want, you can use table 15.3 as a guide, because that gives you the differences, and you can look up the differences in both the figures. You guys get the numbers I gave you? Okay. Go through the table and see if you can see those differences, what they are listing between the sympathetic and parasympathetic. Use this table and compare these two pictures. Okay, don't get lost with anything else. Then we'll be here for the whole day. Okay. How can you think of something more practical to think of your whole nervous system, your autonomic nervous system? What can you think of? Okay. Highway. Okay, what is highway? Is it your brain, spinal cord, your skull?
It's all the nerves. It's all going to one.
Yeah. Then how do you differentiate your autonomic system from that? Good question. Okay, I'll give an answer and let's see if it's good or bad. Okay. You have this whole college campus. They have a master control for the lights, for the temperature. Okay. And let's assume, I don't know if it works that way, each room has its own control. Each building has its own control. So by turning on the main switch, all the lights come on, all the AC, everything works. But also, each building, each room can have its own control, right? If the temperature goes up in this room, the other room is fine. If there is a way, I mean, some buildings do have that, you can adjust within your room. Like in the car, you have, some cars have 4 controls, right, for your AC, or something like that. So it's the same system, the same wiring, the same nerves, but some branches go into these different organs in a specific way, so you can turn on, turn off. So the sympathetic increases everything except the digestive, and there are some more functions it will inhibit. I don't see that in the book, like kidney and liver also it will inhibit. And the parasympathetic does the opposite. It slows down things or lowers everything except the digestive system. Okay, that's as far as how it works. But you have a list of differences on page 571. So what's the first difference between these two? Origin in the CNS. So what do they say for the sympathetic? Thoracolumbar versus craniosacral. So can you see that?
Yes.
So all the branches of this sympathetic nervous system, I'm going to say SNS, otherwise I'll be tired by the end of the class. SNS, they come from the spinal cord, but more from your thoracic and lumbar region. Can you see that? And also, you see parallel to the spinal cord, you see a series of ganglia. They call it sympathetic chain ganglia because they are in a chain. There is another name if you see in the page across or in the description you'll find paravertebral ganglia because it's parallel to the vertebral column. So sympathetic chain ganglia or paravertebral ganglia. But they again synapse with another ganglion. Each one has a specific name. But in general they call them collateral ganglia. So you have the parasympathetic or sympathetic chain ganglia, and you will see the collateral ganglia. But if you see the parasympathetic division, the PNS, you don't see the paravertebral or the chain of ganglia. You see only the collateral ganglia with specific names. Can you see that? Okay. They even call it terminal ganglia if it is very close to the organ. So location of ganglia is another difference. So read the table and see if we can understand those differences. And the third one, what's the third item?
Fiber lengths.
Fiber length. They are talking about the two fibers, the pre-ganglionic and post-ganglionic. So in the first one, the sympathetic, you have a short pre-ganglionic fiber, and from the first synapse, you see a long post-ganglionic fiber. Can you see that? And it's the opposite in the parasympathetic. You have a long pre-ganglionic and the post-ganglionic. Sometimes you may not even see it very well unless you look carefully. It's close to the organ or on the organ itself. Take a look. Look at the branch of the vagus nerve in the parasympathetic. Where do you find the post-ganglionic fiber. It's almost on the heart. Can you see that? Okay. And then the divergence. Divergence means the branching. It's extensive in case of the first one, sympathetic, compared to the second one. Divergence meaning the number of branches starting off from the first synapse with the ganglia. Because if you see the towards the end, you have many branches in both. The effects of the system, sympathetic is more generic and widespread. Parasympathetic is more specific, targeted, and it's local on that particular one organ. So you need to know these differences. Then in the next page, they talk about the adrenal glands. You can see figure 15.6 where the adrenal glands are located above the kidneys. And also you can see in the middle the parasympathetic ganglia. In the adrenal gland, if you see they have a cross section cut open. You see the middle portion and the outside portion. The middle portion is your medulla, and the outside is the cortex. The medulla is one that secretes your adrenaline and noradrenaline, which we also call epinephrine or norepinephrine. So that much you need to know for that adrenal gland. The location, what they secrete, and which part of the adrenal gland, whether it's the medulla, in the middle, or the cortex. Then, now you have this wiring, okay, all these nerve branches coming and making the sympathetic, parasympathetic system to the target organ. It goes through the specific ganglia, right, to the different organs. Now, how do you turn it on, turn it off? How do you make the signal strong, weak? We need what? Neurotransmitters, receptors for those neurotransmitters. So there are specific receptors for certain kind of neurotransmitters. So you can have turning on, turning off of these different systems with the help of the type of neurotransmitter, the type and the amount. In the same way, receptors can be specific based on what kind of neurotransmitter. You can have different types of receptors, and you can have more or less receptors. So that's how you modulate the strength of the signal. So let's see how they compare the parasympathetic and the sympathetic. Parasympathetic, if you see the first synapse between the two nerve fibers and the second synapse with the target organ, what is the neurotransmitter in both? ACH. And what is the name of the receptor on the target cell? Muscarinic. Okay, like muscadine grapes, muscarinic. The name comes from some toxin they studied when they found these receptors in some animal system. But you don't have to worry about it. But always the receptor for acetylcholine is the muscarinic receptor in the target cell. But if you see the first synapse on the autonomic ganglion, you see that's nicotinic receptor. So nicotinic and muscarinic, these are the two types of receptors that can recognize acetylcholine. So you have it in the description under acetylcholine. They call the fiber that secretes acetylcholine cholinergic fiber. And the receptors, they are either muscarinic or nicotinic receptors.
Do you have to remember all this?
Yes, ma'am. That's why I'm explaining to you, I'm giving you the time, I'm asking you to look. You should have studied before class and you should have used the time to look up something. Okay, otherwise everything is Greek and Latin until the day of the exam. Even if I explain any better, it's not going to get in anywhere unless you make an effort. Then if you look under the sympathetic adrenergic fiber, you have two options. One, the part B, you can see the first synapse, you have the ACH released, and the post-ganglionic fiber releases norepinephrine. And the receptor here, what's the other name for norepinephrine? Noradrenaline. And what do they call the receptors? Adrenergic receptors. Okay, so you can relate that to adrenaline. Something you have to relate. First of all, you have to know there are two nerve fibers involved, and what is your synapse? How many synapses should be there? So if you didn't understand the previous one, then we cannot understand this. So we are looking at this under the assumption you use those 5 minutes I gave to look at something. Okay, and I did explain the basic differences between the skeletal system and the autonomic system. In one you have only one nerve fiber, in the other one you have the two nerve fibers, the autonomic ganglion, and the two synapses where you have the transmitters being secreted. So here they are just discussing the differences. So you can see the parasympathetic, you have ACH in both synapses, and the receptor names are different. The first one is nicotinic and the other one is the muscarinic. Both recognize ACH. And in case of the sympathetic, you have either adrenergic fibers on the target cell or the muscarinic. It depends. So you can have both. And there are two types of adrenergic fibers. If you look under norepinephrine, under the subheading, you see the adrenergic fibers and adrenergic receptors. They have alpha adrenergic receptors and then beta adrenergic receptors. But then, if you look through the muscarinic and nicotinic receptors, in case of the nicotinic receptors, as soon as the acetylcholine binds, signal transmits, always. So it's excitatory. But the muscarinic receptors, the receptors are of two different kinds. So depending on which one binds, your target cell is either turned on or turned off. And the same way with the adrenergic receptors, you either turn on or turn off based on whether you have the alpha or beta receptors. Alpha receptors are excitatory and the beta receptors are inhibitory, meaning they turn off. And table 15.5 shows you the various effects of both these systems. That you don't have to memorize, but eventually you have to know. I mean, for your test, I'm not asking you to memorize that, but you have to know which one turns on, turns off. And last but not the least, you have two more aspects. One is dual innervation, and the other one control without dual innervation. So dual innervation is very simple. Okay, nothing is simple by the way, right? So you see the figure 15.9. What do they have? How your iris, how your pupil, how it dilates, how the iris muscles open and close the pupil. And you see one division of nervous system in blue, the parasympathetic, and the yellow sympathetic. Okay. Sympathetic, if you look down close to the place where it ends on the iris, the yellow, what it says? Stimulation of pupillary dilator. Okay? So the sympathetic system, it dilates your pupil unless it has to close. So when it has to close, the other system triggers the constriction of the pupil. So you have two systems involved working against each other. So they call this antagonistic effects. So you see the word antagonistic effects for that description under the dual innervation. First of all, what is dual innervation? What is antagonistic effect and what is cooperative effect? Cooperative effect, you need both of them working together instead of opposing each other. Okay. So if you see the example in the next page for the cooperative effects, they talk about salivation. So when you salivate, the parasympathetic division stimulates salivary glands to secrete a watery enzyme-rich secretion, and the sympathetic division stimulates the mucous cells to produce mucus. So you need both. So you have both the systems, the sympathetic and parasympathetic, instead of working against each other, doing two opposite functions, they are doing the same thing. So they call this cooperative effect. And in some cases, the last aspect, you don't have two nerve fibers coming to work on a particular part of the organ. So here we have, what do we have? A blood vessel, and what are the two actions? Constriction and dilation of the blood vessel. Unlike the pupil, here is the blood vessel. So you have only one nerve fiber coming, but still you can do two things without the need for two branches. How is that possible? What is the difference you see? See the barcodes on the top and the bottom on the nerve fiber? What is the difference you see? You see nothing, then I can go home. Saves you time and
I have
time.
potentials.
Yeah, the signals are much closer in one versus the other. So if you see in case of vasoconstriction, you get high frequency signals coming along the nerve fiber. Okay, that tells the blood vessel to do one thing, here to constrict, right? And if it's if you have more gap, then it does the opposite, the dilation. So it's the same nerve fiber, but depending on the signal strength, frequency, you can either do one or the other. So you can have two lines going to that organ, or you can have one line going to that organ, meaning the nerve fiber, but depending on the signal, you can either turn on or turn off. Okay? If anybody can find a better way to explain to me next class, I want you to come up here and I want to listen to you. Because unless you can relate some information, unless you can make an effort, nothing will get in. Okay? And we have the exam coming soon. So anything you don't understand, you need to stay back and ask me so I can explain to you one-on-one with the little time I have. I mean after class. Or you have to make an appointment with me if you don't understand. But we will never understand unless you go and do your part. After you do your part, if you still understand, I'm here. Okay? We cannot call Ghostbusters on the day of the exam. No anatomy busters. Anything you need to bust, let's do it before the exam. Right? We'll see you.